Welcome to the Conversational Flow, a KCAS Bio podcast.
Hello, and welcome to the 22nd episode of the Conversational Flow, an official podcast of the KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry space for over 14 years.
Thanks, Brian. We are joined by a special guest today. Karla, would you like to introduce yourself?
Hi, I'm Karla Moncada. I'm on the business development team at KCAS. I'm the flow cytometry specialist.
Thanks, Karla. Looking forward to our conversation today. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our headquarters are located in Kansas, with additional locations in Europe and Australia, partner location, we're both here in Philadelphia. Jeremy is in Missouri and Karla is in New York, so we're truly all over the map. We're amped up to have you listening to our 22nd episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're ecstatic to have you here and looking forward to a fun episode today. To kick us off for episode 22, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got an agenda that's not packed, but quite full, I would say, of cell therapy news and cell therapy investments and things like this for today.
Totally new ground for us.
Just brand new things, right? But I always like to follow the field because I feel like they've run into some roadblocks in some directions, and I'm very interested to see sort of how they're going to squeeze out of it in other directions. And this dual target CAR T, that's the first medical research news story, is one of those ways, right? So this is a news story from PenMed. It's about a phase 1 trial that they're running right here in Philadelphia, where they're using CAR Ts targeting both EGFR and IL-13R alpha 2.
And I was joking at the outset about it that, right, it's something that is actually newer for what we talk about because of all the hematological malignancies typically, right? We're targeting CD19, for example, some of them BCMA. However, here what we're looking at is a CAR that's looking at solid tumor, specifically for a glioblastoma, which is an aggressive brain tumor. And so why is it novel if we're going for solid tumor? Well, CARs have generally not been as effective against solid tumors. And the driving force behind that is the tumor microenvironment, specifically, T cells kind of get exhausted. The tumors bind PD-1 on the surface of T cells that causes exhaustion. And so typically what you would see for something like this is what's called an armored CAR, essentially knock down PD-1. Some of the other things that make it difficult for solid tumors are lack of unique antigens or antigen loss, right? So there aren't ways to specifically target these tumors. There may be physical barriers, and then the cells have limited persistence and proliferation just because of hypoxia, the milieu of the area in between cells that really makes it difficult for the cells to survive.
Yeah, so I would say the 2 to 3 barriers for CAR Ts in my mind are a functional barrier in terms of this solid tumor function,
Hmm.
be a cost barrier, just in terms of the cost of manufacturing them and how personalized they need to be, and things like this. And then C, I would think, another a functional barrier of, how easy is it to find a target and actually tune the response, if you will, based on the need of the indication. So this is definitely targeting one of them by going after solid tumors. Interesting to see it in CSF instead of in the peripheral blood.
Yeah, it's certainly notable. I think too here with how aggressive this type of tumor is, what you're seeing really is just an extension of. I'm going to say extension of life. Essentially, we're looking at people up to a year here, patients, I should say, hoping for the tumors to not expand, right? But I mean, it certainly was more effective than no therapy alone. But I think it's certainly demonstrating the hurdles that CAR have had to go through to be able to become effective in this arena.
That's it. So definitely starting off with a very aggressive case to show some sort of improvement there, and we're optimistic that these types of techniques and these different strategies that you mentioned in terms of armoring them or changing the targets will really lead to breakthroughs to break down one of these three barriers. Really, so optimistic about that.
Yeah, and
The Sorry,
go ahead.
go for it.
I was going to say, I think it's really interesting. You had mentioned about the barriers and how CAR are made is one of them, right? Getting the cells out of the patient and then having to modify them, put them back into the patient. And so here, your next item really lands on specifically changing that.
That's exactly right. In terms of this immune onkia result from ASCO, correct? In terms of this next gen CD47 antibody.
Right, exactly. And so, right here we have a dual therapy, right? We've got an anti CD47, and then it's paired with lenvatinib, right? So we got a small molecule kinase inhibitor. And so essentially trying to combat the growth of tumors while also, utilizing CD47, excuse me, which modulates phagocytosis and activation of B and T cells. And so trying to enable the immune system then to attack these tumors.
That's right. And of course, antibodies, as you mentioned, have the benefit of being able to be manufactured centrally, distributed, and ideally have some sort of stability. And they're also definitely not patient specific. So certainly a more easily scalable. The industry's had 50 years head start to really make this a viable approach. So again, can reach more patients more quickly from that standpoint. And with these kinds of response rates, definitely we're optimistic about that.
Yeah. And I do want to jump in. I was trying to do a lead in here, and evidently I failed because I was thinking about your first item for the investments that's actually in there. But still, the point about treatment here, this is antibody, right? Antibody driven plus a small molecule. So separate from CAR. But certainly it's mentioned here 2 or 3 partial responders resistant to first line therapy. And so this, a lot of what they're talking about here is using AI driven biomarker analysis, AI powered digital pathology analysis, right, to be able to understand how the treatment's working. Specifically, here they said that the high CD47 tumor levels essentially had 60% objective response rate versus no response for those with a low CD47 count on tumor cells. And so, I think, again, one of the common themes we've been talking about in podcasts here is the use of AI, and you're seeing that come to play more.
That's it. How can you make the medications more and more specific, or make sure that you've got let's say, hundreds or thousands of medications for a given indication, how do you make sure to select the exact right one for that patient and that disease state? So ideally, this gets closer and would open up a path. It may be difficult for drug developers who then need to collect all this information as they're running the clinical trials in terms of which patient types respond best, but it gives them a window to make sure that they can, still maintain some sort of respondent group instead of, potentially finding a negative group if the population size is too big or not delineated by these biomarkers.
Yeah, yeah. And I got to give you props, Brian. I threw you a curveball there, and you just maintained calm and steady as ever. So I certainly appreciate your steadiness there.
That's what we aim for. But yeah, I mean, in terms of the segue, though, definitely one of the biggest things that's happened in the recent past year is AbbVie's purchase of Capstan. I think this, I now see exactly what you're referring to, because this Capstan purchase is exactly related to that manufacturing comment I made about how expensive it can be to create CAR Ts using the most well-known sort of autologous process.
Yeah, exactly. And so this is what I was referring to, where they're using lipid nanoparticles to deliver messenger RNA. So we've talked about this before, but reminder, messenger RNA, very famous for the vaccines that we saw for COVID, specifically the Moderna and Pfizer versions. And so here what they're doing is getting that messenger RNA to the cells that need them to be able to reprogram the T cells, essentially doing what a CAR T program would do outside the body. They're doing this inside the body, which certainly reduces the cost of production there, makes it a more amenable process. And so another thing too, you're talking about here mRNA, but a lot of times when you're doing gene therapy or something, you would be utilizing lentivirus or adenovirus, and you generally can't do repeat dosing because the body will have had some sort of response to it. Here, it's mRNA, but lipid nanoparticles, there is much less of a chance of something like that. And so the side effects are more mild in that respect, at least as it relates to the delivery there.
Sure. I mean, for anybody that's gotten multiple doses of a Moderna COVID vaccine, then that's basically what could be expected here instead of the much more severe sort of antiviral response.
Mm-hmm.
Now, that being said, when you generate CARs, there's always the chance of cytokine storm and things like that from that going on. But I would imagine it'd be significantly less if you're putting in, you're really not putting in cells. I imagine you're generating many fewer CAR T cells as well.
Mm-hmm.
But definitely an interesting technology. And again, the impact of targeting those nanoparticles to specific T cells would make them just much more specific. I know the problem with LNPs in general is that they just all go to the liver, and you can slightly influence that to slightly target other organs as well. But these are actually specifically targets that they can get taken up when they're in the bloodstream by their target T cells. So ideally, it would significantly decrease the dosage if it is well targeted like that.
Well, not only that we're talking about T cells, but specifically CD8 positive T cells, which I thought was pretty impressive. Then on top of that, as it being an mRNA, is transient, right? So once the message is, essentially translated, then you no longer have the mRNA present. And so at that point in time, you aren't proliferating these CAR cells, right? They do their job and then they're done. And so there are times where it's beneficial not to have that persistence versus other times we've had studies where you would want to have that persistence.
That's it. And then also, I guess, the benefit of doing this RNA approach, it would be significantly faster to try to iterate and look at different targets and things like that if needed. So that can be definitely a great thing for AbbVie as they move forward with this technology to try a whole bunch of things all at once, as opposed to needing to spend years developing a single therapy or a single strategy for how to develop these cars.
It's funny though that when I read this article, in my head, I'm like, we talked about this already. Why are we doing this again? And then I saw you get your note here about the AstraZeneca acquisition with Esso Biotech, which is doing something very similar. And so that's what I was thinking about. So yeah, growing competition here in this space.
Yeah, and basically the differentiator between these companies. So there's Esso Biotech, there's of course Capstan, but there are also companies like Umoja and Interius, and basically each of these are using different targeting mechanisms for their in vivo CAR T, and I think that's really going to be the differentiator there.
Absolutely.
Yeah. But a huge investment in the CAR T space, which is always good to see. Huge investment potentially in the autoimmune space as well, or immunology in general, or it could go into oncology still as well. So definitely good to see that kind of cash being thrown around in the biotech space, especially for 2025 here.
And I mean, the other side of it, you're talking about the cash being thrown around, is this makes it ideally potentially more affordable, right? Because of the manufacturing not being as costly.
Well, that's it. In terms of patient impact, like this overcomes a lot of the barriers that I would have. Like if I had an autoimmune disease and it wasn't, severe, like debilitating impact on lifestyle, I would struggle to justify either the cost or the risk of a real CAR T infusion in some cases. Whereas this could decrease it to the point that it becomes, a manageable risk or cost. So that would be the dream, right? That autoimmune strategy of tuning your immune system instead of taking, completely foreign drug would be brought to life there.
Yeah, and so now what I'm going to do is I'm going to leave the lead in for the next one to you. Got to leave it to the professionals.
Fair. So for the next one, the other story we've got today is also on sort of this advanced drug targeting modality theme. And here we've got a story that caught my eye about Chimera and their IRAK4 program.
Yes.
And they paired this weirdly with a new partnering discussion with Gilead too. So I found that interesting.
Right, and so they had been, working with Sanofi, so the lead candidate KT-474, but identified that another candidate showed enhanced selectivity and potency, and so actually shifted gears there. Expected to enter clinical trials in 2026, so be interesting to see how that progresses through. What Chimera is looking at is something essentially called molecular glue. So, degrading targets specifically to achieve some sort of result, right? They're breaking the pathway down so that you're not seeing, specifically here, the trials are for atopic dermatitis and hidradenitis suppurativa. And so, yeah, it's really interesting technology, and obviously one that Chimera is obviously able to play up for large pharma, both Sanofi and Gilead, working with them on this technology.
Yeah, and the reason I include Chimera and this protein degrader technology in general is because if you think about how drugs have previously worked, for the longest time, it was just the drug needs to bind to something on a target, and that binding itself needs to be the action. In recent past, then we've been able to significantly expand on that so that the binding to the target can now basically bring cells into proximity and have cells take action, or it can bring a virus to target a specific cell, and that can take action. With ADCs, the binding can bring a drug molecule again into proximity that can take action. This protein degrader technology can, instead of just bind, it can actually take action on the target itself. So that's where I find the technology very interesting, and I think that's why they're leading to so much success with these larger companies that can apply this technology, where they probably have in their pipelines all sorts of discovery targets that they said, oh, that's great, we can bind to it, but we can't do anything about it, and now they can. So it's interesting to see that being applied across the space here.
And to our point at the outset, I was joking around about CAR and having talked about that a lot. This is something that we had brushed on previously, but hadn't really had as much exposure to in the podcast. So it's good to see new technologies coming to the fore.
That's it. Yeah. And then, of course, in terms of KCAS news, the big thing to announce, and especially flow cytometry related, of course, is that KCAS has now successfully and completely implemented Elastigate in terms of a machine learning capability.
Yeah, this is fantastic. Really looking towards the future of machine learning and trying to streamline processes. So what does Elastigate do? You can essentially feed it data. It learns from that data how the gating is supposed to be done, spits out analyses for you. Now, obviously, with that, it's not one of those things where we just trust machine learning. Not that there's a problem per se with machine learning, but want to make sure, just with anything AI, right? Whenever you go into ChatGPT, it says ChatGPT can make mistakes, right? So we've got QC, Tech QC behind Elastigate to be able to ensure that the analysis is done properly, but it saves time in going through the process, and really also I would say aids in consistency there, right? Because the broad strokes, everything is getting done up front, and then it's just that fine tuning at the back end when we do the Tech QC. And so really excited to see this coming together.
Yeah, I mean, that word implemented is doing a lot of work there, right? It's not just plug it in and see if it goes and cross our fingers. But definitely, this is, I think, the way of the future for flow cytometry, and whether it's this algorithm or other algorithms that we're also evaluating, then I see a lot of the analysis being done there. I see it opening up new opportunities for analysis as well. So definitely very excited, and kudos to the team for making that happen.
Absolutely.
The next portion will go into our sort of paper of the month and sort of main portion of the talk on global harmonization. But I did want to bring our guests back into the discussion to really bring this out and give a chance to introduce her new video series as well. So, Karla, did you want to take a minute and more fully introduce yourself and talk about the new Karla's Corner series?
Yeah, I'd love to. Thanks, Brian. I just started a video series called Karla's Corner, and really what I want to do here is try to connect with the network a little bit more. It's really difficult to connect with people when you work remotely, and definitely going to meetings is helpful, but I feel like LinkedIn gives you a platform where you can just have constant conversations all year long. So that's really my main purpose here, just trying to connect deeper with a network and have ongoing conversations about flow.
And now your background, you had mentioned you've operated a flow core for a long time. And then following that, you get to work with some of the bigger companies, equipment companies, and play with, I think you said in your first video, play with the toys, right? And so you want to talk to some of your background there?
Yeah, that would be great. So I joined a flow cytometry core facility in San Antonio, the Health Science Center in San Antonio, in 2006. And at that time, I really didn't have any experience in flow cytometry. I was coming into a brand new technology, and I was just excited to learn. But it was a really cool experience for me. I felt like, In college, I had customer service type of jobs. I worked in restaurants. I worked in a cosmetic counter for a while. So I really liked the interaction with people, and I really liked the science. And the Flow Core let me do that. It let me do customer service in a science setting, right? So I really fell in love with the position. I fell in love with being able to help lots of different labs at the same time. As you know, in science, it takes a really long time to get some good results going. And when you're in a core and you get to work with a lot of people, kind of like what we're doing now, right? You get to celebrate some successes along the way as you have lots of people that you work with. So I was in the facility for 15 years, and I got to work with Ben Daniel when I was there. Ben Daniel is now at Regeneron. But that was part of starting my network in cytometry. When I was at the Health Science Center, I started attending flow techs. Getting involved in their organizing committee there was really helpful to my networking. And then I started being able to attend cytos, which really opened me up to the rest of the community. And it was just, I was able to learn so much by connecting with the flow cytometry community broader, right? So I just felt like the more people I could connect to, the more I was able to connect people to each other when they needed a solution as well. And in 2021, I decided to join SciTech. And at that time, SciTech was launching their Aurora CS. So I was leading the teams in training on the CS, evaluating it internally and externally, training customers. And then I joined the reagent team for a year, and I was managing the reagent team in the Americas. And then I found myself at KCAS this past November. So I'm really enjoying being at KCAS. This is new for me to be in flow cytometry in drug development. So flow cytometry in academia and flow cytometry in drug development definitely has overlap, but not completely. I'm learning a lot.
Okay, and I think that theme of connecting lots of people then fits directly into the main topic in the paper of the month. So for this month, it is around harmonized phenotyping of human, I'll just call them MDSC for pronunciation ease there, but MDSCs by flow cytometry results from an interim study.
Right, myeloid-derived suppressor cells. There we go.
Okay.
And so this is in cancer immunology, immunotherapy, impact factor of 4.84, right? And so they're looking at identifying these cells. Now, why are they important? Well, MDSCs are implicated in knocking down the ability of the immune system to be able to combat cancer, to be able to kill tumor cells. And so by immunophenotype, by monitoring this cell type, which is not just a single cell type, right? There are subsets to it. You can then get a good idea on prognosis and progression. And so what they were hoping to do was invoke all these labs to see how they were doing it and see how much consistency there was.
That's it. And that's why I say lots of people. It was interesting when I was preparing for this podcast, I wanted to find a good example of an international collaboration that was focused on standardization. And I found that actually relatively difficult to find in PubMed. I don't know if I was just searching really badly, but every collaboration I found, all the authors were in one country. So when you start reaching across multiple geographies, there are definitely challenges, which leads to the first sort of main topic question. What are the challenges of working with labs in different countries and or regulatory systems?
And I mean, some of the things that you're looking at are differences in reagents, obviously availability. And so that if they've got a different way that they may identify lineages, that's another thing, different processes, different equipment. And so here, to your point, they were across 23 international labs. And they tasked the different labs for identifying the various lineages, and they saw within the lab there was consistency, which I mean you might expect. But then when you step out and compare lab to lab, the inter assay, they saw much less consistency. And interestingly, one of the things that really drove it was the presence of a live dead discriminator, right? A marker there. And then gating, the analysis portion, were the two real big things that drove it. Now, when they were talking about panel design, there were a significant number of markers, essentially 32 that they had seen in the survey, but only 7 were really core in identifying the MDSCs. And so they did task, what was it, 1 million PBMC per test, and they tried to normalize the MDSCs against the count of the lymph plus monos. So really tried to take some of the variability out of it where possible.
Yeah, but if you don't harmonize on the panel itself and you don't harmonize on the gating strategy, then it will be very, very difficult. There's 100 different ways, especially when you start to get as far down as subsets of MDSC panels, populations, that you could get to the same answer. And if you go down different routes for each one, the numbers won't match. That's for certain, but the numbers will also be telling you slightly different things in a lot of cases. So can definitely see the challenges there from not starting in the same place. And that's why, of course, we always start with what panel do you want and how do you want it gated before we move further, right? But you brought up a couple good points. I had started from regulatory, and definitely, like, if the FDA says do it one way, we will absolutely do it that way in the US. Whereas if the EMA says do it another way, then it has to be done that way in the EU. So then every once in a while you come to impasses, even in the way that you name things, or the way that you have to report things, or who has to sign off on those things. So that can definitely be a regulatory hurdle. But then I thought the next point you brought up that sort of ties directly into Karla's background as well is really around the availability of instrumentation reagents in different places.
Right. And also being able to harmonize instruments themselves. So I know that there's definitely push for harmonizing instruments across sites. And some companies are going out and doing it manually, where they're trying to match up the instruments in-house as much as possible. But once you ship those instruments and they get installed, things do move around a bit. So you still are having to have the engineers on site realigning everything and making sure that the settings are set up to harmonize those instruments across sites, right? There's other companies that have the math doing the harmonization for them. And it's a lot more hands-off, but it's also technology that's a little bit more expensive than the manual technology in the field.
Sure. So yeah, I mean, from the instrument setup portion, if you're not, especially with flow cytometry, where like things just are not as standardized, as you say, it's a lot more manual. If you don't start off with that end goal in mind, then there's no way to keep the different labs in sync. But then on top of that, after you have the instrument installed, let's say you did have the same instruments, but now all of a sudden you go to purchase your reagents for the panels. In different countries, oftentimes the catalog numbers or even the formulations themselves of the reagents can be quite different.
Yeah, and another thing that you want to consider when you're using reagents is making sure that you're QCing the reagents before you're putting them into your precious samples. It's something that not everybody has in their practice, but I strongly recommend it. Mistakes are made. Sometimes things are put in the wrong vial. Sometimes something's a little bit off. So definitely QC your reagents in the lab before you're putting them into your precious samples.
And then as far as availability is concerned, one of the notes that they had in this paper was the clone used, right? So for CD124, that was one of the 32 markers from the survey, and noted that the difference of clone used there impacted variability. And then how they they made up the lineage cocktail. So one of the things they made were the CD3 141956, looking at those core markers to try to remove those cells from the analysis. These things are more impactful sometimes than you imagine. I know in the past we've had discussions with clients when looking at clones for the same marker, and you're getting different percentages. Then the question comes out, well, which one's real? And I mean, it's they're both real. They're binding the way they're binding. It's just there's different epitopes, right? And so yeah, it's always an interesting discussion.
It's like the blind people looking at or touching the elephant and saying what they think, and the one is touching the tusk and the other is touching the trunk, and they think they're seeing two different things, but it's actually one, right? So
yeah, that's a good point. I like that.
Yeah, so even with different instruments and potentially with different reagents, I would feel okay in some cases with being able to discriminate bimodal populations and get a decent frequency alignment between labs. It's really the edge cases where it's not a strong bimodal, or there's insufficient resolution between positive and negative populations, where that gating would become difficult from a percentage perspective. And then from a clinical trial or a bioanalysis perspective, definitely the quantitative assessment in this type of scenario would be not feasible. So definitely from a process standpoint and analysis standpoint, would have to look at the context of use there, and it would to be able to get those quantitative readouts, you need to go to like a next level of harmonization to really talk about the process, make sure everybody's cocktailing the same way, staining in an optimized procedure the same way, and making sure that there's training to propagate that through, because I'm sure this also took place over some period of time as opposed to just like a week or two.
It's interesting. I'm thinking, Karla, to your experience, and I'm wondering what your thoughts are here, having run Flow Core, right? And you've got different groups, different scientists constantly coming through there all the time, which, they have different ways that they utilize the instrumentation, they prepare their samples. And so it's kind of like a microcosm of what they're talking about in this paper, all within one core.
Yeah, it's really interesting to see, how different people are dealing with their cells. Of course, in a core facility, and I was in a cancer center, you're dealing with a lot of different cell types and a lot of very specific needs that they have. So it's not something that I could necessarily always have recommendations for, but best practices is where we can all start, right? And really in a core facility in the academic setting, a lot of this is learning. So having somebody come in with something that maybe hasn't been prepared in the best way isn't always a loss. It's a great place to just learn and say, okay, what did we do here? What can we do better next time? And this is really where you build those deep skills as a scientist to be able to really comment on a broader range of trying to harmonize and follow best practices.
Nice. And then I guess the same for data analysis too, right? Like after you've acquired the data, you've done the learning or the difficult or hard to undo learning, let's say, and now you've got data analysis where you can learn in a much cheaper way, because worst case, you can always just go back and start from the beginning.
Right. Make a new gate.
That's it. So, whether that's being done consistently between sites or centralized or automated is definitely that next level portion there. And then on top of that, once you have the data, is it named consistently so that you can actually interpret it appropriately between sites? Well, it sounds like they like 7 populations or 11 populations that they could do, or 7 markers. But what about the rest of them?
7 markers, 10 populations, right? And so we've already talked about this. Part of the KCAS news was doing Elastigate, right? Machine learning. And so hopefully standardizing, the aim would be standardizing there the analyses, but then on top of that also centralizing it, to your point, and so always doing the analysis within the same core group, so that you don't have nearly as much variability if it was distributed among different sites all the time. And so, yeah, trying to take that next step there and remove as much variability as you can.
Yeah. Yeah, but overall, an interesting paper. And again, kudos to the group for working across geographies there. As I said, there were not many papers that really covered this. So, difficult to get everybody aligned on what needs to happen and really execute a project plan across so many different geographies and priorities and regulatory bodies. So kudos to them. And, it's an inspiration for clinical trials to think about this as we think about any biomarkers that might be needed for downstream testing.
Well, I mean, we've even seen the challenges here when we go to harmonize our own sites within the company, the communication that's required, the harmonization on SOPs and so on and so forth, to ensure that all the training that goes into it, to make sure that you're getting consistent results out of the samples from a study that's run across different countries, across different sites. And so you could imagine then taking that to that next level where this is outside of company bounds. The challenges are just so much higher.
That's it. So yet again, that word implemented is doing just a lot of work here. Maybe we need to come up with a new word or phrase that can do some more work, or sounds like it's doing more work.
I think you got to frame that word on your wall in your office or something. I think you like that word.
Implemented, indeed. Yeah. All right. Fair. All right. Anything else to cover on the paper?
No, just certainly appreciated it like you did. A lot of effort that goes into something like this. It's not easy, and I think it certainly advances the knowledge of the field when you can see what are the pitfalls of something like this, and everyone can learn from how to implement harmonization properly. And to a larger, we talk about SolCap all the time, right? The standardization, how much it's really necessary. We just keep on hammering that point home, I think, podcast after podcast.
That's it. And towards that thought, then I'll go to fan feedback and ask our fans to comment on what aspect of a validation procedure changes the most between different assays. And I ask that because for an immunophenotyping assay, it's relatively straightforward, right? In terms of H62, you have to do your intra and inter assay and some stability assessments and a few other test scripts, but it's relatively prescriptive. Whereas for RO assays or different functional assays, there can be a lot more questions in terms of what are you interpreting as your base state? How are you maintaining as your base state? So that's really what this question is getting at. What do you find that you have to tweak for each validation to make them fit within your paradigms?
It's a good question. Hopefully get some good feedback there, some good answers from our listeners.
That's it. In terms of other questions that may elicit some feedback, of course, now is the banter section. So my big thing for our July episode is what coping techniques do you use for your pets around fireworks?
I have a feeling you have background on this, but Karla, why don't you get us started here?
Well, I will say my dog's never had a problem with fireworks. But my sister does have a pretty neurotic Vizsla. His name is Chase, and he's on a hefty dose of Prozac. And for fireworks, she will also supplement with a quarter of a Delta 8 gummy.
Oh, wow. That's awesome.
It worked really well for him. He is a happy dog.
There you go.
So, for me, I've got 3 cats, and the amount of chaos that they cause doesn't seem to change at all. when there are fireworks or if there are no fireworks, they run around the house and destroy things just as much either way. I've got scratching posts, I've got toys, there are places for them to go and play. One cat, for some reason, loves to chew everything. He like the curtains at the bay window that we've got. There's a huge section of like 2 feet by 3 feet that is just cut out because he chewed on it so much. And I think he's trying to expand beyond that so that he has better visibility. I think he has something with curtains. And then my other cat seems to be so happy to just jump in the way when I'm trying to feed them in the morning. I got one of my cats is diabetic, right? So I have to feed them on a regular schedule. I just can't leave food out because he will literally just eat himself constantly. And so I got to prevent that. But the other two, I want to call them kittens, but they're 2 years old now. High energy. The second I get this food, they're jumping up on the table and knocking stuff out of the way. So they cause their own fireworks, I think, Brian.
Okay. So no modifications needed. I would always have to put mine in like a super quiet room and like close all the blinds and curtains and things like that. But never needed more. I've heard of, as Karla mentioned, drugging them with puppy Xanax or whatnot, or I've seen like something called thunder vests as well.
Yeah. I've seen that.
Mm-hmm.
But
so talk to me about these thunder vests. I've heard some about it. It's like a nice hug, right?
That's it. Yeah. Just a constant pressure that makes them feel like they're being coddled, like they were in, back in some sort of den or something like that.
And it's
I've heard about them.
Go ahead.
I've heard about them because I had Italian Greyhounds, and they're pretty neurotic dogs to begin with. So this is a common tool that you can use to just kind of soothe them, to get them to calm down a little. I never had to use them on mine. Mine just jumped all over the place, and I didn't try to mitigate it too much.
Now, isn't it the same principle though as weighted blankets, for example? Right?
It is. And I was going to bring that up. I tried a weighted blanket one time. I found it to be terrifying. Like, I felt like somebody was like holding me down or something like that. But I don't know. And maybe that's just me, not a pet. So
you're the first person I've heard who has said no to that. I will be honest, I don't have a weighted blanket. Like, I'm the kind of person who sleeps really hot. So, I as few blankets as I possibly can. But I had always been curious about that and if it does enhance sleep. So I appreciate you starting to balance that ledger, because everyone else I know says, oh, it's the best thing ever.
It is pretty cool. I've used a weighted blanket. I sleep really well, so it's not like I need the help sleeping. Mm-hmm. I know that affects some people. But yeah, I sleep really well. I tried a weighted blanket and it was just fine, but I didn't like that I couldn't just easily kind of move at night and bring it with me. I really had to like wake up and pull it with me. So it wasn't a good go for me.
Okay. That's 2 no's.
Yeah. All right. Good to know.
Yeah. All right. But that does bring us to the end. So I do want to always thank our guests for listening. Please send any feedback, requests that you have, or any tips or requests for new episodes, and we're happy to accommodate.
Yeah, definitely want to thank all the listeners that have reached out on LinkedIn and any other platform, honestly. But always great to hear from you, and just let us know what you're enjoying about it, what you'd like to see, anything that you'd like to see different. So thanks, everyone.
Yeah. And for our next episode in August, we're tentatively planning the title as "What Happens If You Go with the Flow Too Much." So looking forward to that. So thanks, everyone, and see you next time.
Thanks, all.